right, so just a reminder that we're talking about macromolecules. These are the large molecules that combine to form cells. Okay? In our previous lecture, we addressed carbohydrates and proteins, and so today we'll finish up by talking about nucleic acids and lipids. Just want to remind you that carbohydrates, proteins, and nucleic acids are all polymers, and they are formed by the polymerization or the linking together of monomers. And remember also that each one of these has its own specific type of monomer, and this chart is shows that also. Okay, so first we'll talk about nucleic acids. Nucleic acids are our third type of macromolecule, and there are two types of nucleic acids. There is DNA, which stands for deoxyribonucleic acid, and there is RNA, which stands for ribonucleic acid. These two molecules are very similar, but they do have some very important differences. We'll be coming back later this quarter when we talk about structure of DNA and gene expression and talk about these in more details. So today will be sort of a preview or the basic information. One thing that is very different about these two molecules is their job or their function. So DNA functions to store genetic information. Hopefully you recall that from our cell components and organelles lecture. The DNA in eukaryotic cells is found in the nucleus, and in prokaryotic cells it's just free floating around in the cytoplasm. RNA is involved in gene expression. Gene expression is the process of taking that information that's stored in the DNA and using it to make proteins. And we'll be talking about that in a lot of detail coming up later this quarter. Okay. Today we're going to talk about nucleic acids and what they're composed of. They're polymers of nucleotides. So what that means is that the nucleotide is the monomer that when you take many of them and link them together, you form a nucleic acid, and that is the polymer. Okay. So let's look at the nucleotides. So there are 8 total types of nucleotides. They do have a lot of similarities, but there are 4 DNA nucleotides and there are 4 RNA nucleotides. Luckily for us, they all have the same basic 3-part structure. And then there's just some differences off of that. Okay. So what is that 3-part structure? Well, first of all nucleotides, so all 8 of them have the same phosphate group, or they have a phosphate group. And they also have a pentose or a 5-carbon sugar, and then they have something called a nitrogenous base. And we're going to look at each one of these in a little more detail, but those are the 3-part structure that every nucleotide has. Okay? So notice that it does have a sugar in it, right? It incorporates a carbohydrate into it. And remember when we talked about the 5 different types of common monosaccharides, we mentioned deoxyribose and ribose as 5-carbon sugars, and this is where we see them being used. Okay, so the phosphate group is always exactly the same for all of the nucleotides, and oops, it should say 8 there. That's a typo. Change that to 8. For all 8 of the nucleotides, it just has the same phosphate group, and remember, a phosphate group is a phosphorus with 4 oxygens. We often abbreviate it with just a P with a circle around it, and we'll see this later also when we come back and talk about ATP, which has 3 phosphate groups. So that's a nice shorthand for it. Okay. The pentose sugar is different depending on whether you're talking about DNA nucleotides or RNA nucleotides. Okay. In DNA, the 5-carbon sugar or the pentose sugar is deoxyribose. Okay. And this is that same diagram that I showed you in our carbohydrate lecture that shows the deoxyribose sugar. And here on carbon 2', remember that we always count these with starting to the right of the oxygen. And then because we're doing a 5-carbon sugar that's incorporated into a nucleotide, we use the little prime. Okay? Oops. So it's not just 1, it's 1'. And on the 2' carbon, there is just 2 hydrogens. There's no OH group. Okay? So when we're talking about DNA nucleotides, we have that same phosphate group, and then this sugar is the deoxyribose sugar, which is different in RNA. In RNA, we have ribose. Okay? And the only difference between ribose And deoxyribose is that on that 2' carbon, we have an OH group instead of a hydrogen. Okay, so deoxyribose, deoxyribose is, D means missing an oxygen, so it's deoxygenated. It's missing one on the 2' carbon. So if we're talking about RNA nucleotides, those 4 RNA nucleotides have ribose for their 5 carbon sugar. Okay, and then that leaves us with the nitrogenous base. Okay, so the base is a nitrogen-containing ring-shaped structure. Okay, some of them have a single ring, and some of them have 2 rings that are stuck together, but they always have nitrogen. They have a fairly large component of nitrogen. They have multiple nitrogens found within the ring. Okay? In your activity that you're going to do in class on Thursday, you're actually going to be building a nucleotide, so that will help you understand that. Okay? So there are 5 different bases. So there's 5 different options to be attached to here. Okay? And each base gives the nucleotide its name. Okay? It's the main thing about the nucleotides that are different. Okay, so if you look at the 4 DNA nucleotides, they have the same phosphate group and they have the same deoxyribose sugar, and then they have each of the 4 have a different nitrogenous base. And we see the same thing for the RNA nucleotides. All the RNA nucleotides have the same phosphate group and the same ribose sugar, and then the 4 different ones have 4 different bases. So let's look at these a little more in detail. Okay, so these are the 5 different bases that are possible. Okay, these structures would replace this generalized base structure that is shown here. Okay, so if it is cytosine, then this structure here, and notice that it has 1, 2, 3 nitrogens incorporated into the ring, but also some carbons and some functional groups hanging off of that. That would be attached here. Okay. We find cytosine in both DNA and RNA, so that means there's going to be a DNA base that has the phosphate group and the deoxyribose sugar and cytosine attached to it, and an RNA base that has the phosphate group, the ribose sugar, and then the cytosine base. Okay. The other 2 that are found in both DNA and RNA are guanine and adenine. Okay. So again, just like with the cytosine, we'll find these bases. These are really big ones, right? These are double ringed. Both of them have lots of nitrogens and 2 rings attached to each other. We'll find that attached where the nitrogen base goes for both DNA bases with the phosphate group and the deoxyribose sugar, and RNA bases with the phosphate group and the ribose sugar. And then there's these 2 that are different. Okay, so we only find thiamine in DNA. So that means we're only going to find the thiamine base attached to the phosphate group that's attached to a deoxyribose sugar. Then we'd find the thiamine here. And in RNA, we have uracil. Okay, and we'll come back and we'll talk about this more when we talk about gene expression. Right now, I really just want you to understand the basic structure of the nucleotides. I'm definitely not expecting you guys to memorize these at all, or to be able to understand the differences in detail between them. We'll be talking about them more later this quarter, and for now I just want you to understand that that's what's sitting here. Okay, and it varies, and it gives the nucleotide its name. Okay, and then here is a slide that shows all that information. Okay, so here are the DNA nucleotides. Often we just call them DNA bases because between the 4 DNA nucleotides, the only thing that's different is that base region, right? They all have the same phosphate group and the same deoxyribose sugar, and then what's attached up here could either be thiamine, adenine, cytosine, or guanine, right? And then that would give that DNA nucleotide its name. Okay, so the one, for example, the DNA nucleotide that has thiamine attached at the base, we call that thiamine. Okay, and then we see same thing over here in the RNA nucleotides. Okay, and the RNA nucleotides, they all have the same phosphate group, they all have the ribose sugar, and then they all have a base attached. And the base could either be uracil, adenine, cytosine, or guanine. Okay, and again, whichever base is attached is going to give that nucleotide its name. Okay, so if guanine is attached here, then we call that nucleotide, we call it guanine. Okay, so these nucleotides, no matter what base they have, are going to be attached to each other in long strands to form a nucleic acid, right? Remember, the nucleotides are the monomers, and the nucleic acid is the polymer, okay? So this occurs by dehydration synthesis, just as it happens for carbohydrates sticking two monosaccharides together, and for putting two amino acids together. We're always connecting these via dehydration synthesis. Okay, so what happens here is that the oxygen, and this should have a little prime on it. The oxygen on the OH on the 3' carbon of the 5 carbon sugar is taken off and combined with the hydrogen from the phosphate group of your other nucleotide. Okay, take oxygen, hydrogen plus a hydrogen, you're gonna get what? Water, right? So water comes off, and remember, that's why it's dehydration synthesis, and that water is shown here. Okay, when the water is taken off, then we see if I can change color here. When the water is removed, then a new bond is made between this oxygen and this carbon. Remember, that corner is a carbon here, and that's what is shown here. So here is the new bond. Okay, this bond is called a phosphodiester bond. Remember, I said that a lot of the bonds have their own specific names. That doesn't make it a different bond. Or, it doesn't make it different than a covalent bond. It's just a specific covalent bond that is holding two nucleotides together, right? Because here was one nucleotide, and now here's that nucleotide still, right? All of that is the nucleotide, and then here is the second nucleotide, and it is attached here through this new bond that was made when water was pulled out. Okay, we call that a phosphodiester bond. Okay, so that makes a dinucleotide where you have two of them together. That's not what, nucleic acid is much, much longer than that. In DNA, for example, there are millions of nucleotides linked together to form the nucleic acid that carries our genetic information. So, but this process just keeps happening over and over again, just like we saw with the carbohydrates to form, say, starch, or the amino acids to form a very large protein. We just keep adding new monomers onto the end. Okay, so here again, here's our dinucleotide that we made before, and now we want to add another nucleotide onto that. And it doesn't matter, these are just shown randomly, it doesn't matter which base it has, the same process happens no matter what, right? So this OH is going to come off the 3' carbon, and this oxygen is going to come off the this hydrogen will come off the phosphate group, and that will go and come off as water, right? So another way to show that, you could show the water over here. A lot of times you see it coming off here as, part of the reaction. It's just a different way to show it. It's the same information. So now I've shown water coming off 3 places. It really only comes off one, right? There's only one water molecule that comes off. So you would pick one of these to show, right? I wanted to show you where the OH and the hydrogen come from to make the water. This is just another way to show it, and then another way to show that you produce a water. And now you've taken that dinucleotide here and attached it to another nucleotide, and this right here is that new phosphodiester bond, right? And so now we have 3 nucleotides linked together. There's 1, right? Here's 2, and then we have our third one here. This is number 3. And this is showing the same thing, okay? So this is part of a DNA molecule, or this is what it's supposed to be. It could also easily be, oh no, it's definitely DNA because it has a T instead of U. Okay, so remember, every nucleotide is going to have a phosphate group, a 5 carbon sugar, and a nitrogenous base or a nitrogen containing base. Okay? And then the only thing that's different as we go down this whole line The only thing that's different is that base region, right? The phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, that stays the same down the whole way. And then we could point out the different parts of different nucleotides on here. Okay, and you guys should be able to do this. So there's one nucleotide, and when you circle your nucleotide, you want to make sure that you include every part of that nucleotide, but not the phosphate group, because that belongs to the next nucleotide. So there's the second nucleotide, the third, and the fourth. And that's nucleic acid. Of course, they're much, much longer than this. All right, our last type of macromolecule are the lipids. The lipids are a kind of a little bit of a catch-all group. It's a diverse group of molecules, and unlike the carbohydrates, proteins, and nucleic acids, they don't have to have a whole bunch of stuff in common. Instead, one of the things they have in common is that they are not polymers. So if they're not polymers, that means there are no monomers. Okay, so we're not going to link together a bunch of monomers to make a polymer, and it's going to be a lipid. That doesn't happen. Okay? And then, but one thing that they do have in common is that they have regions of many carbon-hydrogen and carbon-carbon bonds. And hopefully you recall that these bonds, because those two elements have equal electronegativity, and of course carbon-carbon have equal electronegativity, they are nonpolar, and that makes the molecule have a significant hydrophobic characteristic. Okay, we'll see when we go through these that not all of them are completely hydrophobic. It's not that they don't say have any oxygen in them at all. They're just mostly hydrophobic or have a huge portion of their molecule that is hydrophobic. Okay, a great example of a lipid that you probably come in contact with is oil. Okay, what happens when you put oil in water is that the oil tries to get away from the water, right? And that's because it's hydrophobic. It has all of these nonpolar bonds. It cannot make hydrogen bonds with water, and so they try to get away from water. They can't dissolve in water. Okay, so we're going to talk about 3 types of lipids, and all of these have that in common. They're not polymers, and they have a significant nonpolar hydrophobic characteristic to the molecule. Okay? So the 3 types are fats and oils, which seem different but actually are very similar and are generally called triglycerides. We're going to talk about phospholipids and steroid hormones. Okay, so first of all, fats and oils. So fats and oils, I'll show you the chemical structure. They don't actually have too much of a difference. And they are both used for long-term energy storage. So in animals, the lipid that is used for long-term energy storage are fats, and another name for that are saturated fats. Okay, and I'll show you why, because we'll look at the molecule. These are solid at room temperature, so things like cheese, bacon fat, eggs, beef fat, all of that stuff is solid at room temperature, and those are animal fats mostly. There are some plants that will also make saturated fats, but mostly it's animals. Okay, and of course we use this for long-term storage of energy, and when we look at the molecule, I'll tell you how that works. Okay. And then the other type of fat we often call oils. Their more scientific name are called unsaturated fats, and again I'll show you the molecule and explain why. Okay, these are primarily produced by plants, although some animals do produce some unsaturated fats, but mostly it's plants, and plants are using them for the same reason that animals use them, for long-term energy storage. Okay, plants will often put these in their seeds for the embryo the plant embryo to utilize when they start growing in the spring. So we often find them in high concentration in things like nuts, or like olives that have a lot of oil in them also. Okay. If you're comparing a fat to an oil or a saturated to an unsaturated fat, one of the biggest tip-off is what state is it in at room temperature. So fats are solid at room temperature, and oils are liquid at room temperature. Okay, let's go ahead and look at, don't forget this. Okay, so fats and oils are very similar to each other. Okay, they only have a small difference, and I'll point that out later. What they do have is the same basic structure. So they have a glycerol head, and that's what this region is called here. We call it the glycerol head. And then they have these 3 fatty acid tails. Change color. And then they have these 3 fatty acid tails. Okay. These fatty acid tails are called acids because they have this carboxylic acid group on them. You recognize that. But the rest of them are purely nonpolar, right? So each one of these CH2s, what that is really showing is carbons bonded to each other and also bonded to hydrogens. Okay. Remember I said that lipids have a significant hydrophobic nonpolar characteristic and have lots of nonpolar carbon-carbon and carbon-hydrogen bonds, and that's where you see them. You find them in these fatty acid tails. Okay, so to make a fat or an oil, a glycerol head is added to 3 fatty acid tails, and then you get either a fat or an oil, which is generally called a triglyceride. Okay, the glyceride part comes from the glycerol, and the tri comes from the fact that there's 3 fatty acid tails. All right, let's look at saturated fats. So saturated fats have a glycerol head, right? So that is shown here, the glycerol head. Okay. So that's the glycerol head. And then these 3 fatty acid tails, okay? They are called saturated because what this is really referring to is that it's saturated with hydrogen. That means it has as many hydrogens as possible and that there are only carbon-carbon single bonds. Okay? There are no double bonds. And when we contrast that with unsaturated fats, you'll see the difference. Okay? And that means that these tails are very straight. Okay? So fats, or saturated fats, have these long fatty acid tails that are straight, like that. Okay? So here's a little cartoon diagram that we can represent it as. There's the glycerol head, and here are the 3 fatty acid tails that are straight. Okay. I also want to point out something. I said that fats are used to store energy, and that's because of these carbon-carbon and carbon-hydrogen bonds. Carbon-carbon and carbon-hydrogen bonds are nonpolar, and that means that the electrons that are in those covalent bonds are shared equally. So you could draw them kind of right in the middle, okay? And that means, that's actually means that they're both tugging on those electrons equally. And that is a situation that actually has a lot of what's called potential energy. Energy that has the potential to do work. It's like 2 people pulling on a rope equally, right? What happens if one lets go, it releases a lot of energy. And that's how the energy is stored here in these bonds, both in the carbon-carbon and carbon-hydrogen bonds, because those electrons are stored equally. We'll come back and talk about that idea when we talk about cellular energy and cellular respiration. I just wanted to put it out there. Okay, so let's talk about why are saturated fats solid at room temperature. It has everything to do with these long fatty acid tails that are straight. Okay, when the fatty acid tails are straight, the fat molecules can get really close to each other. They can pack in very tightly so that there's not a lot of space in between the molecules. Okay, and that means they don't wiggle around a lot and they behave as a solid. Okay, I just want to point out to you, just remember, we do see fat molecules associating with each other, but they do not form any covalent bonds. Okay, so this is one saturated fat, and here's another saturated fat, and another saturated fat, and so on, but they are not monomers. Okay, they are not linked together by covalent bonds. They are instead hanging out with each other because what's surrounding that usually is a bunch of water, and the water, or the hydrophobic tails, want to get away from the water. Okay, so if there's water molecules everywhere, that is going to make the saturated fats clump together. So we get clumping, but no actual covalent bonds between them. Okay, let's look at unsaturated fats. So unsaturated fats are liquid at room temperature, and they have a similar structure. Okay, they also have a glycerol head. Okay, that's shown here, and then they have these long fatty acid tails, but they have a significant difference, and that's right here. Okay, remember with the saturated fats, we have only carbon-carbon single bonds, and that makes the tails straight. With unsaturated fats, we have at least one carbon-carbon double bond, okay, and that causes the tail to bend or to stick out, okay, so it causes a kink in the tail, okay. Unsaturated fats have to have at least one carbon-carbon double bond, and it's unsaturated because in order to exchange a carbon-carbon single bond for a carbon-carbon double bond, we had to take off those two hydrogens that would be there normally, because remember, every carbon can only make 4 covalent bonds. Okay, so we've unsaturated it with hydrogen. We've taken away some of the hydrogen. Some unsaturated fats may have more than one double bond. It could have many. It could be polyunsaturated fat and have many double covalent bonds, or it could be monounsaturated fat and have just one. Either way, it makes it behave as a liquid. Okay. So here's a little cartoony guy of that. Here's the glycerol head, and here are the fatty acid tails, right? A couple of them are straight. Again, you could have them all have kinks in them, but I just showed it here with just one kink. And this kink right here is caused by that carbon-carbon double bond, and that means that it, it's like has its legs sticking out. Okay, and to understand why this is a liquid at room temperature, it's all about that kick out, right? So the molecules just can't get as close to each other as they would if they didn't have that kink in them or that kick out. Okay, and so they still associate with each other, but they don't get as close, so there's a little more wiggle room. That means they can move around more and behave as a liquid. Okay. A really good analogy with comparing the saturated fats to the unsaturated fats is imagining if you took a bunch of people, say, and put them in the elevator. Okay, if everybody stood with their legs straight down and their arms straight down, you could fit a lot of people, or a fair amount of people in the elevator. You could cram them all together. Of course, everyone would be packed really tightly together, right? Then they would all be saturated fats. If instead you tried to put people into the elevator, into that same elevator, but instead ask them all to stick a leg out, okay, and to not get close, not interact with each other's legs, like you couldn't get that close to each other, then you could fit fewer people in that same space. So it's less dense, and that's the same thing that happens with the unsaturated fat. So they still hang out with each other, but because they have that kick out, there's this space that forms in between the molecules, and that allows them to wiggle around some more. Okay, but again, we still just have them associating with each other or clumping together because of the water that's everywhere. Okay. And they don't make any covalent bonds between them. Right? They're not monomers. All right, next we're going to talk about phospholipids. Phospholipids are the major component of all biological membranes. So when we talked about like the plasma membrane that surrounds the cell, the organelle membranes like around the mitochondria, around the nucleus, around the Golgi, around the Rough-ER, around the vesicles, all of that stuff is primarily made of phospholipids. And I mentioned this when we talked about the vesicles, how they bud off of the Rough-ER and go to the Golgi, and then bud off the Golgi and go to the cell membrane, and how they can do that because they're all made of the same stuff. Okay, and we get traffic going towards the cell membrane and traffic going away from the cell membrane. So phospholipids are really important. Okay, this is a phospholipid. Phospholipids have some things in common with a fat, but not everything. There are definitely some significant differences. I'm gonna get faster at this. I just know it. Okay. So the phospholipid has in here, it has a glycerol head hidden in here. Okay. And it has some fatty acid tails. These ones happen to be shown as unsaturated, and most phospholipids do have unsaturated tails. Okay, but notice that there's only 2 fatty acid tails instead of 3. Okay? And then the other really significant difference is attached to the glycerol head is this whole group here. Okay, that has a phosphate group. Phosphate groups are always negatively charged, and then it has this this hydrogen, or this positive charge on this nitrogen as well. Okay, so we end up with this head region that is charged. Okay, it has a positive and a negative, and that makes it very polar or very hydrophilic. Okay, so phospholipids are an interesting type of molecule. They have down here. They have a portion of the molecule that is very hydrophobic, okay, and that's starting really here. Okay, so we have these hydrophobic tails. Okay. And then a portion of the molecule, and it does include this carbon double bonded oxygen, a portion of the molecule that is very hydrophilic, so hydrophilic heads. Okay. A molecule that has 2 natures is called amphipathic. I don't expect you guys to remember that. I just thought I'd throw it out there. So just like we drew a fat little cartoony guy, we can draw one for a phospholipid as well. Phospholipid has the polar head, or the hydrophilic head, and the nonpolar tails. Okay, remember it only has 2 of the tails, and they are usually unsaturated, so have at least 1 double carbon bond. All right, so let's talk a little bit about how they are used to form cell membranes. Okay, in a cell there is water everywhere, right? Come on, there we go. So in a I did not want that. Sorry, guys. In a cell, there is water everywhere. Okay. So when this molecule interacts with water, say there's some water molecules here. Okay, water. Maybe a better way would even be to draw an actual water molecule, right? So if we have some water molecules here, okay, the 2 different regions of this molecule are going to behave differently, right? The polar head is going to want to make hydrogen bonds with that water, right? So we can make a hydrogen bond there. And so it will want to interact with water. But what about the nonpolar tails? They cannot make any hydrogen bonds because there's only these carbon-carbon bonds and carbon-hydrogen bonds. Okay, so the nonpolar tails act like oil in water. Okay, and remember the oil in water tries to get away from it. So that means the nonpolar tails try to get away from the water, and the polar heads try to get towards it. Okay, and what this forms is called a lipid or a phospholipid bilayer. Okay, these naturally form when you take a bunch of phospholipids and you put them in water. They will automatically form this bilayer structure where you have 2 layers of the molecules. Okay, because there is water out here, say this is outside of the cell and this is inside the cell, the polar heads face the water while the nonpolar tails form this hydrophobic inner layer. Okay. And this is what it looks like in a cell membrane. This could be organelle membrane or the plasma membrane of an entire cell. Both of them are have this phospholipid. Let me write that up. Phospholipid bilayer. Okay, and that's what this is here, right? With the hydrophilic heads facing into the organelle or into the cell and facing out. Okay, of course the inside of the cell is full of water, and surrounding the cell is also a bunch of water. Okay, so those hydrophilic heads face the water, And the hydrophobic tails don't like the water because they can't make any hydrogen bonds with it. So they instead face the inside and form this hydrophobic inner layer. Okay, this understanding the structure of the phospholipid bilayer is important when we start talking about the function of cell membrane. So remember, the cell membrane is like the gatekeeper, or it gets to decide what goes into the cell and what goes out of the cell, or into the organelle and out of the organelle. And that's all about this structure. Okay, so we'll, in our, I think it's our next lecture. In our next lecture, we'll be talking about that. So keep that in mind. Okay, finally, we're going to talk about steroid hormones. Okay, steroid hormones are different than the fats and the phospholipids completely. We don't have any fatty acid tails or anything like that. But remember, those characteristics of the lipids is that they are not monomers and they are nonpolar hydrophobic. Okay, and we see that here. The one thing that they do all have is this whole set of 4 rings, 3 of which are 6-sided, 1 of which is 5-sided, and those are all linked together, and then with other stuff linked to it. Okay, each one of these is a carbon, right? Remember, the carbon is just shown with a corner. Okay, so there's a lot, and then there's tons of hydrogens hanging off of this, and then it has these hydrogens here, so they're definitely nonpolar. Okay, and this is one molecule. This is one cholesterol molecule, which is a type of steroid, 1 estrogen, 1 testosterone. There's no monomers that form these. It just is a giant molecule. Okay. I don't expect you guys to like have a super deep understanding of cholesterol, estrogen, and testosterone, but I just wanted to tell you what they were, just so you had an understanding. Okay, cholesterol gets a bad rap. There's definitely good cholesterol and bad cholesterol, but cholesterol is really important in the cell membrane. So in that phospholipid bilayer, cholesterol is stuck in there, and it helps increase the fluidity of the cell membrane. So that's one thing I didn't talk about previously. I'll just bring this back up. Because they have unsaturated fatty acid tails, right? The phospholipids actually act as a fluid, right? And it makes the cells, be able to give and take. They're not hard. They don't have a hard surrounding. They have a soft fluid surrounding. That allows vesicles to pinch off and that kind of thing. Okay. So also added in there are also these cholesterol molecules, and that helps maintain the fluidity. Cholesterol is really important, particularly in brain and nerve cells. Right. And then there are some sex hormones. There's estrogen. So this is estrogen. It's the female sex hormone. Don't try to memorize these things or anything like that. I'm definitely not going to ask you what the effects of estrogen are. I just wanted to put a slide up here just in case you had no idea what kind of things the female sex hormone did. Now you can see what they do. But estrogen, triggers the creation of female characteristics. Right? And the male sex hormone, testosterone, triggers the creation of male characteristics in a human. Okay, and so this is testosterone. Testosterone and estrogen have some striking similarities, right? They're not all that different from each other. And indeed, they are both made in a similar sort of way. All right, that's the end of that lecture.